Welcome everybody to the TDWG 2021 virtual conference, and in particular to the Symposium number 8, entitled From the Field Right on Your Science Table, Challenges of Sharing Ecological Data. I am Yanina Sica, and with Paula Zermoglio, will be the co-moderating this session. And we're grateful for the tech support of Avery Bender and all the University of Florida conference team. First of all, thank you all for joining, and we especially thank our speakers in this session. Just as a reminder, this session will be recorded and available in WOA for later viewing. And each speaker will present for 10 minutes, and there will be 3 minutes of questions at the end of each presentation. I would ask you to please ask all the questions to the speakers using the Q&A feature in WOA, and Paula or myself will ask the questions to the speakers. And the chat function here is available for any technical questions. Of course, please use it wisely. And bear with us with any technical difficulties that may present during the presentations. And I will ask all attendees and the speakers when they are not presenting to mute their audio to avoid weird things. And can you put the name slide, Paula? Now Paula will give a very short introduction, and then each of the speakers will have their 10 minutes. So thank you, Teresa, Xiaojun, Diana, and Peter for sharing your work, and please enjoy the session.
Thanks, Yanina. So, well, I'm just going to mention some things just as a very, very brief introduction to our session. And I will start asking all of us, what do we think about when we think about ecological data? And so some of us may think of populations, for instance, an animal population of that little endangered frog. Some of us might think of communities. Some of us might think at the landscape scale. On the other hand, there are scientists and others concentrating on what nature can provide us, so ecosystem services and food production, and then there is diseases and distribution of organisms that transmit diseases. And so ecological data is a very big, very heterogeneous term, let's say, that brings together a lot of different kinds of data and a lot of different kinds of people working with those data. So undoubtedly, we associate ecological data with nature and with how we interact with it, and probably sometimes what we can get out of it. Now, of course, that is related to decision making at every single level. So it's decision making at the individual level, at the political level, at the scientific level, and all in the public sector, in the private sector, and all over. And as I was saying before, this involves a lot of people that are in the community of scientists and in the large community, and involve citizen science people, and it also involves, as I said, the private sector and many others who are contributing data and ways to analyze those data. And so we find there that those data are collected in the field by very different people, and those data are analyzed and tools are built to interpret those data, and collaborations are built, and we're linking those ecological data with other types of data. We cannot interpret data about organisms without putting it in a context, for instance, of the climate or the environment in a particular place. And so we're linking that to environmental variables and sometimes to social variables like landscape use or resources use. And so we see that the landscape, allow me, for around ecological data is very, very heterogeneous. And so we think of different types of data, of different scales or granularity. We think of different data providers and users, which each have their own needs. And we think of different uses. And of course, we think of different standards and tools to work with those ecological data, right? To be able to share it. And allow me, yes, to use this already cliche word, maybe, that is integration. We need to integrate very different things, very different types of users and providers and types of data. And so we need to talk to each other. And with that idea in mind, we put together this symposium, and we have the set of talks that Shani was mentioning before, which involve pieces, elements that are working with ecological data from different perspectives. So the idea is that we are all at the same table, and we're all trying to find common solutions to use this ecological data for preserving or conserving nature, but also for using it through its ecosystem services. So I invite you to stay in our session and enjoy the different aspects that contribute to ecological data and help us discuss how to best integrate these different approaches. So with that, I will give the floor to Teresa. Thank you.
Hello, thank you. I will share my screen with you. Just a moment, please. Is it correct?
Yes, it's perfect.
Hello, everyone. I would like to thank you for the interest on my speech on this session. And I also thank the organizers of the event. It's a pleasure to be here again. My name is Teresa Cristina Yanina, and I work at the Instituto Tecnológico Vale in the north of Brazil. This institute is a non-profit multidisciplinary research institution. And we are developing a project that's called the Biodiversity 4.0, Standardizing Biodiversity Protocols for the private sector. The private sector has increasingly recognized the need to achieve environmental, social, and corporate governance. The acronym for this is ESG, through measurable indicators and data survey and storage. So the idea of data is a key idea in this process. So we are working in a tropical forest, maybe the National Forest of Carajás, inside the Amazon biome. And this poses a special challenge because Amazon harbors a rich biodiversity. And we have also working with traditional knowledges and the local communities. So it's a really challenge to incorporate, to integrate all these different types of data. And we are working with standardized biodiversity protocols in order to help us in field surveys. And this standardized data can help in decision-making process, especially related to risk management, uncertainty reduction, avoiding data loss, sharing public data, also predefining the surveys themselves, to conduct long-term monitoring, to use innovative technology, and to design nature-based solutions for the region. We have these 3 lines of action. We are working with digital standardization of the data. long-term monitoring, and automatic acquisition based on innovative technologies. We want that our framework is based on the principle of fair data. That means the data is findable, accessible, interoperable, and reusable. We are integrating very different types of data in an integrative, standardized virtual database. And we are also relying on Internet of Things, location intelligence, and artificial intelligence for this. Just to give you an idea about the different types of data and the different topics of ITV research related to biodiversity. You can see on the right figure the different types of lines of research that we are conducted inside our institute. And on the left side, you can see that we have mapped for now 366 terms that we included inside the 22 classes, and we are now comparing for the with the Darwin Core protocols. But we know that some of our data will not be covered by Darwin Core, so we are now defining new metadata to our specific needs. We also conduct an interview with scientists here in Brazil that led long-term monitoring projects. And these are the 6 main ideas that they emphasized. Consider biodiversity, you have to think about the key data and the key standardized data in our case, the key permanent plots that will be surveyed. Also, considering the governance of the project, we have to have clear aims and also a regular budget, especially thinking about long periods of time. We have to have also an engaged team because people change during long periods of time, and the survey has to continue. And also the engagement of local community is a key point because they can help us to a better effective, most effective surveys. We are also using automatic data acquisition. We are using certain devices for automatic data acquisition. Recently, we acquired a LiDAR and a drone that we will be used mostly to get functional traits data. Because the vegetation in Amazon forest, the canopy is very high and the understory is so close, so it's very difficult to do this kind of surveys without this type of equipment. We also have hydrometeorological stations because we are interested in data related to water resources and also to local weather, local climate. We are using data logs and also camera traps. In the right side, you can see a photo of a big cat inside the forest that we captured with one of our camera traps. We are using also tags of RFID, mainly to study the movement of bats and bees inside the forest. And we are also using sound records. And with this last device, we captured most of them. We already identified most than 400 species, species of birds. So you can see that it's a useful approach. Our main aim is to build a positive legacy in the region. We are mapping and protecting the services provided by the ecosystems. We are working with agroforestry, food production, and the mailing pollinators, the role of pollinators in the food production. We are also studying the role of the forests in the purification of air, carbon storage, local climate regulation, the protection of water resources, the protection of soil health, And also we are working with local communities. We have a biofactory of stingless bees to enhance the awareness of the importance of protecting pollinators. And I think that is all. Thank you. Maybe I go too far. It was too fast.
No, that was perfect, Teresa. Very interesting presentation. I remind everybody to use the Q&A tool in WOVA to ask their questions. So if anybody has a question, please. We have 2 minutes to answer any questions. If nobody has one, I can start because I have one. So that was very interesting, and thanks for mentioning the need of standards in taking this type of data. And I've noticed that you were saying that you had like more than 300 fields, like different fields of
Yes.
metadata. So later, I don't know if you will be there, but later we will talk about standard framework for this type of inventory data. So it will be very interesting to keep having this conversation with you and see if we can adjust all those terms. But now generally on your presentation, I wanted to ask you, so you're also involving people from the community to make this surveys, or is it people working for the company? How is that?
Both things. We also have a partnership with local communities because the environment is very challenging. So it's necessary to stay there with people that know this environment better than us. I live in Belém, that is the capital of Pará State, and our field work is 800 kilometers from here. It is inside the forest. So local people knows much better than us, the region, the places. So you are working with them very close.
Wow, that's very interesting. And great pictures in your camera traps, I have to say.
Thank you. It's a great place, yes. Thank you.
I can imagine. Perfect.
We do have a question in WOVA.
Yep.
I think we have time for one more. It says, you might have said it, though, how do you store your data? Do you have an integrated data infrastructure at the Institute?
It's not my field of expertise. I am a biologist, but yes, we have a group of applied computing that is working with us, and also people that is specialists on database, considering the technology, the information technology. So we have a cluster inside the ITV. And for now, we are integrated the data there, but I don't know, maybe at some point we will need other types of equipment. But for now, it is enough to us.
And there's another question related to storing and sharing. That is, Teresa, are you storing the standardized protocols where they can be reused by others?
Yes, this is our aim. We want that our data and our metadata will be shared with other people. And we are at the beginning of the process. This project just initiating this year. So we have a long path away, and we are in the start of the process. But yes, we would like to be to share the protocols and also the metadata with the other people and the public itself.
And we have one more there, and we pass to the next speaker. What is your experience like to get the necessary permits to conduct ecological research and collect data?
Permits. It's not so difficult. Here in Brazil, the main organization, governmental organization, is the ICMBio, and we have a close contact with them because the National Forest of Carajás is a protected area. So they are very aware of all of our activities inside the forest, and we need their support for this. So this is not so difficult to have these permits because we have a longer, the ITV has 10 years already. So we have a long process of this kind of partnership with governmental or organizations that protect the forest. So it's not so difficult. I'm not sure if I understand correctly the question.
Well, we'll see if more comes up in the Q&A. So just a reminder, these questions will be there for everyone to see later, so you can revisit it in the platform. Thank you very much, Teresa. We are going now to pass to our next speaker. So it's Xiaojun Wang.
Yeah, I'm going to share my screen.
Thank you, everyone. Thank you.
Thank you. So Xiaojun Wang is going to give her presentation about HydroClim Data Portal, Cyberinfrastructure for providing high-resolution GIS model streamflow and water temperature data to researchers.
Oops. Okay, can you guys see my screen?
Yes. Ready when you are.
Okay, I'm ready. Sorry, my voice is sounds like weird today. I must say sorry first. Okay, hi everyone. I'm Xiaojun Wang. I'm a software developer from Tulane University Biodiversity Research Institute. Today, I'm going to talk about the HydroClim Data Portal, a tool, a cyber infrastructure for providing high-resolution GIS modeled streamflow and water temperature data to researchers. So we know the freshwater ecosystems is playing a key role in sustaining aquatic biodiversity. The environmental changes has also had significant adverse impacts on water temperatures and streamflow. The changes in temperature and precipitation forecast next century are expected to affect the freshwater ecosystem and their biodiversity. HydroClim project is aimed to generating the liking basic hydrological data, which is describing variation in streamflow and water temperature across North America. So in this project, we define this streamflow and temperature as HydroClim data, and we hope this project can increase the public understanding of water resources issues and provides more training opportunities for conservation of freshwater biodiversity in future. So first, we need to generate the data. So Dr. Jason Loft from St. Louis University and Dr. Darren Ficklin from Indiana University and their teams has generated the first initial batch of raw data using the soil and water assessment tool, hydrologic and water temperature models. We can call it SWOT models. They generated two groups of data by the SWOT model. The first one is the predictive simulation data from 1950 to 2099, which is produced from multiple representative concentration pathways scenarios, where we can call it RCP. It have two standards of 4.5 for 19 models and 8.5 for 20 models. And these models are used in global climate model scenarios too. It will produce the runoff and soil erosion and water quality for each ridge segment. The ridge that is a river and the ridge in the watershed of all across the United States and Canada. And the second group is the historic SWOT data, which is comparable with RCP models data between the historic time periods. You know, the historical measurements are spotty and they are insufficient locations and time periods for the historic water temperature and stream flow data. So this SWOT historic data can fill these gaps. They used graded historical climate observations as input and produce the data in the same format as RCP models data. So we can see what main channel output files looks like. I didn't include all output files here because there are 10 to 20 files generated by the SWOT models. This is the most important output files. So we can see there are 57 variables here, and each row represents 1 month of stream data for each segment. And with in these files, the most important variables is like the ridge number, the time step, the flow, stream flow, and the water temperatures. So the next question is, how could we extract, host, publish, and share this HydroClim data with the scientists? So Tulen team is setting up a Cyberinfrastructure to extract the HydroClim data from the complex SWOT model output files to reintegrate the data and build a data portal for the users from both temporal and spatial dimensions. Roughly, we have 4 main parts of it. We have R scripts for data extraction and import, and a GIS database for hosting 1.9 billion HydroClim data over 37 watersheds spatial data. And we also have a web-based API to query and download the HydroClim data and spatial data. We also have a data portal interface for users to let users download and query and visualize the HydroClim data. So here's the data flow. First, we need R scripts to extract the temperature, stream flow, and spatial data from the SWOT outputs, watersheds, and watershed shape files. Then import them to the Postgres database, which is hosting on the database server. So how users can access the data from database, which in that case, we set up a web server, which including a web-based data portal interface and the API. Users can send the data request to API through the interface. Then the API will wrap the required data and send it back to the users. Users also can access the visualized HydroClim map through the data portal. The map services are provided by a GIS server. It uses open standards to publish data from any major spatial data source. I will show you how to query and download the HydroClim data with the data portal interface. So this is the interface for downloading the data. When users access this page, they can select the date, the time period, and what watersheds they want to access. Here, I only select one month in January 1950, and I also select 3 basins here. Step 2 is to choose the models. So users can choose historical data and or at least one RCP model. The next step, the final step is to choose if the user want to download the raw data or the statistical summaries of the HydroClim data. Here I select the raw data and the statistic of HydroClim data in average values. Then the user can click download your data and of the request. So once the users download the required data, this is what's gonna come up for them, which is a zip file. It includes the raw and statistical monthly and the simulated streamflow and temperature data and watershed spatial information. This illustrated with the structure of the zip file, which is going to be show you the different choices that you have picked before, or the different requests you have made. So how can users to use this HydroClim data? For instance, users can use the HydroClim data as environmental data in species distribution modeling to predict the habitat distributions. And I'm also thinking about if the Darwin code can be extended to include HydroClim data for fish and other aquatic organisms. And these data are also easily to be imported in the GIS application like ArcGIS and QGIS.
2 minutes.
Okay, it's a screenshot from QGIS. You can see the HydroClim data has been drawn with watershed layer by rich segments. But we also, the HydroClim Data Portal also provides an online visualization too. We have visualization options, which you can pick either the temperature or the flow, your date, and other parameters in the previous data query page. In this case, you can visualize the stream flow data of the rich segments of all watersheds with the RCP model in a time period. And this is for visualizes the temperature data. This map is still not interactive now. So we are going to let users can query and download the data from this map too. We also have another plans on this project. So we are going to add in data portal for the other United States and Canada watersheds and integrating HydroClim with FishNet 2, which include the occurrence fish data containing geospatial information and following the sharing principles of the globe of the GBIF, and providing a tool for visualizing the ecosystem diversity based on the merged data of HydroClim and FishNet 2. This is the FishNet 2 access API. This is a simple demo of the integration of HydroClim data and FishNet 2 occurrence data. You can see the fish trap channels are distributed in water with a temperature of 22 to 25 degrees. Okay, this brings me to the end of the presentation. Well, I'd like to thank everyone on the acknowledgement side who had helped us in this project. I sincerely appreciate your interest in and attention for this presentation. Thanks.
Thank you very much. Go ahead, Shani.
Yeah, no, thank you. What a great presentation and amazing tool for people to get environmental data on watersheds. This is impressive. Again, everybody who has questions, please use the Q&A tool. I do have a question. It's more like I'm very interested in how the user for your integration with FishNet. I think that's a great integration, right? To get to use that environmental data for species distribution analysis. So could you tell a little bit more about that integration with FishNet? I think the last slide was very visual.
Yeah, well, but that is under progress.
Yeah,
we have a plan for that. But because we have the FishNet, we can download the FishNet from we can download the fish occurrence data from the FishNet API. We also have a HydroClim API. We can integrate them together by like the programming or something because it's web-based. So we can try to follow some standards or principles to integrate these two data together and to extension more possible opportunities to do the research in the biodiversity research like that. I think it's based on the technical
Yeah, very interesting.
Mhm.
Paula, I'm not following the Q&A. Do you see any questions?
No, there are no questions so far. People must be thinking and incorporating all this information.
Yeah, of course. We can leave also some minutes at the end of the session for people to ask questions and free for discussion.
Sure, yes, we'll have a discussion time. So thank you very much,
Thank you.
Yao-Jun. We will leave the floor then for Diana Bowler, who is going to speak to us about modeling the heterogeneity within citizen science data for biodiversity research. Diana?
Yep. Yeah. Hi, everyone. Good morning or good afternoon, wherever you are in the world. Let me just share my screen with you. That should do it. Let's see. Okay. So I hope is that okay for you? Can you see my screen? Yeah.
We can see it, but with your notes.
Okay. Hang on. I know what I'll do. Stop share. I don't need that screen. Okay, it should now be better.
There, perfect.
All right. Yeah, so I'm going to talk about my experiences of using some of these.
Diana, sorry.
Yeah?
I think we're seeing it cut. We don't see the full title there.
I don't know what's going on. Maybe share screen. Now?
No, it's okay. Perfect.
Okay, sorry about that.
Go ahead.
Yeah, so I'm working on a project called SYM08, in which the long-term goal is to examine biodiversity trends in Germany, so changes in species populations and distributions through time. But to do that, we need to first compile a lot of data. In Germany, like a lot of other countries in Europe, there are a few national coordinated biodiversity monitoring schemes, but for most taxa there isn't. But there is activity happening, but it's more local monitoring, regional groups collecting data. So if we wanted to work at the national scale, we had to first bring together this scattered data across Germany. And also bear in mind that this data then hasn't been collected with any uniform protocol. We had to develop ways to synthesize these data to make reasonable biological inferences. So one group that I've been working a lot on is dragonflies. This is a nice case study for us because there isn't a single uniform scheme for dragonflies, but there is a lot of natural history groups collecting data across the country. So I've been working together with, especially with the German-speaking Dragonfly Natural History Society, the GDO, but also with other conservation agencies and different groups collecting data. So some of the people I'm working with are on the left here. And on the right is showing the distribution of the data that I have. So this data is very simple data. It's simply species occurrence records. So it's simply a record of a species being seen on a given date in a given place by somebody. So we've been able to compile data pretty much over the whole of the country. As you can see in this map here, there's some hotspots in the darker colors, but it's a pretty good coverage. And the right shows the time series of the availability of data. So unsurprisingly, data increases in amount through time. But yeah, we have some data going back to 1980 onwards. So that's the time frame of our study. So I'm not going to talk a lot about the stats, but many people have been interested in developing methods to make use of this unstructured data collected without a single protocol. So we're following the lead of other researchers on this and using occupancy detection modeling. And these are quite attractive in many ways for modeling this data because they're so-called hierarchical models. So they allow for the fact that the data is generated by two processes. One process is the ecological or biological processes affecting the species. So land use, habitat, climate, these all obviously affect where species are found. But these occupancy detection models also include a section or a submodel for the observational processes. So what affects how people are collecting the data and how does that kind of act as a filter for what we have in our database at the end. So as I said, several other people are using these models. Other people are also finding they give results that make general sense, and we find that as well. So this is just a quick summary of our results applying occupancy detection models to that dragonfly data set that I mentioned. So we had 77 species in that analysis. If we just look at the top graph here, so the y-axis here is a long-term trend. And this is the zero line means there would have been no change in the trend of that species, trend in its distribution since 1980 onwards. So these yellow bars refer to increasing species, and the purple bars refer to decreasing species. So one advantage of the way we're working with SMON, we have all these experts working with us across the country, sharing data and also expertise. We've been able to do a good check on whether the predictions are making sense, and they do seem to be. So the model seems to be doing a reasonable job. But so it seems to be going okay, but we can suspect that we can do better, and that our model is probably biased in some way. And this is because we know that occupancy detection models, well, they have various assumptions, but one of the assumptions is that there's no unmodeled heterogeneity in that observational process, in that detection process. And we also know from, simulation studies that when there is unmodeled heterogeneity, then the occupancy estimates will tend to be negatively biased. So we suspect this is probably going on in our analysis. So I'll tell you what we can do so far with our detection model or observation model. And then the next slides, I'll talk about what I would like to do with the ideal metadata. So based on the metadata that's currently available in my data set, like I said, it's very simple data. We just know the species, the date, the place, and often we also have some recorder ID. So we just have those columns usually in our data sheets. So like others, we're trying to model some of the variation in the data with proxies of efforts, so survey efforts. We can suspect that people looking for spending longer time looking for species will be more likely to find a species. So we want to include effort in our model. And this proxy of effort that we're using is so-called list length. So that's based on the number of species that somebody reported on a given visit. So a visit is a survey on a given date to a given place. And you see in the data set that actually most list lengths are of just a single species. Most people, when they submit a record, are just submitting a single species. So the biggest division is actually between list length of one versus a longer list length. And most likely these submissions were of list length of one, probably do not represent a complete survey. Most of the time when people are going out and doing a true survey, they will at least see more than one species in most habitats. So that's our proxy of effort at the moment. We can also put in proxies of observer expertise. So we can use this very simple metadata to calculate things like the total number of observations that someone submitted using that recorder ID information. And if we make the assumption that people with more expertise are submitting more records. It might not be the case, but might explain some variation. Then we can at least include this in our model. So that's what we're doing so far. But yeah, we know that people are varying in lots of ways, and ideally we'd include a lot more of this in our model. So the various ways in which we know people vary. So for instance, people will vary in how they choose places to go looking for species. Do they prefer forested areas or open areas? Are they looking in the trees, or are they looking on the ground? All these things will vary among people. People, I have to speed up. People also vary in protocol, so whether they're actually doing a formal survey, or are they just incidental observations that they're seeing while they're doing other stuff. Survey effort, people will spend different lengths of time, and people will vary in their ability to identify different taxa. So I did a survey last year to try and understand more of this variation, and I don't think I have time right now to discuss it. But this survey showed all the different ways that people vary. So it showed, for instance, that most people are doing a combination of different types of surveys. So they do some planned surveys. So this hotspots here. Most people are doing many active planned surveys where they go out specifically with the intention of looking for species, but they're also doing some opportunistic data collection as well. And when they do a planned survey, they do tend to record all the species that they see. That's what this purple bar is showing here. But when they're just recording opportunistically, they're more likely to just record a rare species. And I'll skip this one. So as a researcher using this data, the metadata that I'd really like to be included in my in the data sets, and that I could then include in my model, the following. So I'd really like to know whether an observation is coming from a complete checklist or not. So are people recording all species that they saw during a visit, or are they just selecting a few that they find interesting in some way? And the reasoning here is that if people do report that it was a complete checklist, we can then infer the absences. So we don't just know what someone saw, but we also know what they didn't see, if we know it was a complete list. Regularized recorder IDs would really help analysis of this data, because the recorder names that I have in my sheet, at least, are very messy, sometimes just initials, sometimes just first names, and that really really makes it tricky to try and model variation among people. And obviously also some measure of survey effort, so time or area would really help my analyses. And actually, so eBird is really, for me, leading the way in terms of collecting this metadata. They're doing all of this already, and you can see how valuable that's been for the research that they're producing. And you can also see that it hasn't slowed down participation by citizen scientists, that they're willing to provide this metadata in these databases. So I think my take-home message is really that, yes, there is heterogeneity, and that itself isn't the problem from a stats modeling point of view. The problem is rather lacking the metadata to model the heterogeneity. So better metadata would allow us to really make more use of these big biodiversity databases. And yeah, there's a line of modeling at the moment called integrated models, which would allow you to model multiple data streams at the same time, which would also be useful in these cases where we have these heterogeneous databases. So yeah, don't be afraid of heterogeneity. We just need to model it, basically. Sorry, I think I went a bit over the time. Sorry about that.
Thank you very much, Diana, for a great presentation. I think we have time for one question. Maybe we have one in WOBA. Read it to you. I'm always puzzled by the strong rise of collection data in the past 50, 40 years. I would have thought that the high times of species observations were much earlier, 19th or first half of the 20th century. why this rise at that point? Is your graph, oops, sorry, something moved and I lost it. Here, why is that the rise at that point? Is your graph showing cumulative data availability or data collected by year?
Yeah, it's data availability by year. I guess, you know, so what we have is just data that's been digitized. There's probably a lot of notebooks hanging around in places and in literature that hasn't made it yet to one of our databases. So I know that that's something that a lot of people are doing right now in Germany, digitizing a lot of these older data sets. But I think still we would see more records in more recent years. You know, citizen science has become more commonplace. I think, you know, many people have heard of it now. There's more apps. It's become a lot easier for people to contribute data in a more formal way than before. So yeah, I suspect there is a lot more older data, but I also still think it's increasing now.
So we have more questions in the Q&A and on the chat. So
oh yeah, there's one here.
Okay, go ahead.
I see one in the, so I don't have the Hoover, I just have the Slack. Carlos, yeah, at the moment so I know there's a database called a database in Germany, which has soil species occurrence records. We haven't got there yet. The next analysis we've got plans are for beetles and for amphibians. But yeah, maybe eventually we can start thinking about soil organisms, actually. I think that this would be a nice database. And it's exactly the kind of data that we're using, the stuff that's in a database. It's very similar to that. So yeah, hopefully in the future. Yeah, a question about whether complete checklists vary over time. Yeah, actually, I get, so the problem is so in my data set with the dragonflies, I don't know which are complete checklists or not. So with eBird, you could start to look at that because they do have complete checklist data, but that's only available for the last years. So I don't know exactly whether complete checklists have changed through time, actually. I could investigate that by looking to see the proportion of the singletons and whether singleton records change through time, because I suspect these singletons, that they're kind of the opposite of the complete checklist. And we could test whether singletons have become more common. But yeah, I haven't done that yet, but I should do, yeah.
Thanks, Diana. We have 2 more questions in the Q&A that maybe we can grab after all the talks are done.
Mm-hmm. Okay, cheers.
Thank you. So we're now passing to our next talk, which is Yanina, Sica, and myself. I will share the screen, Shani.
Yep, go ahead. Thanks, Diana. That was a great presentation. I have a couple of questions also, so let's talk about in the discussion part. Okay, so Paula and me, Yanina, we are presenting our talk entitled Unlocking Inventory Data Capture, Sharing, and Reuse. And we'll present the Humble Core extension to Darwin Core, a work that we have been doing over the past few months. Can you go to the next slide? Sorry, I'll keep asking you to change slides.
No worries, no worries.
Okay, so as we know, there are multiple types of biodiversity data. I will talk particularly on 2 types. One that Diana described very well in her example for the dragonflies, which are incidental records, where a person, either by direct observation or using some methodology, records a species in a particular point and in space and time. And these records lack a taxonomic scope, so it only provides, most of the time, only presence information, presence of a species. In contrast, next slide. In contrast, inventories have a defined taxonomic scope. For instance, the list of butterflies or dragonflies in New Haven, where I'm speaking. And having that taxonomic scope enables identification of non-detections, so we can know which species are not recorded. Of course, that will be composed of true absences and false absences. And depending on the duration and the sampling effort, one may increase the presences and decrease those absences. Here we briefly show 3 types of inventory data. One is the elementary inventories that address a single sampling event. Extended inventories combine multiple single events, so we can have a larger spatiotemporal scope. And summary inventories that represent a greater aggregation and involve potentially multiple sampling parties or different methods. And we will talk about different aggregation of inventories later. But the important thing is that this data provides presence and absence information for a species. So basically, inventory data are records of multiple species at a defined place and time. And this provides high quality ecological data because it enables that inference about absence of species in that area and in that time. So they are particularly relevant to assessing and modeling biodiversity and its change. However, their reliability for using those models depends on knowing or having the information about the inventory process underneath it. So the scope, the methodology, and the associated sample effort and completeness, which are often either unreported or described in an unstructured manner, making it difficult to reuse and interpret. Next slide. So what needs to be described about the inventory process so that we make a good use of this information? First, we need to know where the survey, where and when the survey was taking place. So a description of the number of sites, an outline or a detailed understanding of the place where it took place and when, or the time frame. Then how was the survey carried out? So data on the methodology followed. So what type of inventory it was, what about the effort, how many tramps or how many people per unit of time were involved in the survey. Also, we need to know who made the survey and where is it published. So a bit of information on the data set and the citation. And also, very importantly, what the survey was about, so the survey scope. And generally, this inventory process produces a species list, which may be empty if you didn't find any species, but hopefully it will have many presences. Next slide. So now we know that the key information out of that inventory process is the who, the what, the where, and the when. And looking back at different biodiversity standards, we see that the Darwin Core currently enables reporting some of this information. For example, via the sampling protocol or the sampling effort terms. However, that information may not be as detailed or as structured as we would need to make a good use of this inventory data. But building from the Darwin Core, we realized that different types of inventories can be thought of as events. And according to Darwin Core, the event is an action that occurs at some location during some time. So it makes perfect sense. So Paula now will tell us a little bit more about how events can be associated with inventories.
Thanks, Shani. So I'm retaking here with event, and I want to show you a very brief example of how we think of events, because this is kind of the core of what we call humble core, right? Sharing event data at different levels. So imagine that you have this is South America, and so we go sample, and we might find one little beetle at a particular time and place. And so we call that an event, finding that beetle, and that event is associated with an occurrence, which is the beetle. We could also find 3 beetles in that same observation event or collecting event, and we can then have 1 event with 3 occurrences associated. Or we could also think about it as 3 separate events. We can discuss that later. But that is for a particular event. Now, suppose those were traps, were pitfall traps. And so we decide to put all those pitfall traps together afterwards, because we decide that, well, we put 10 pitfall traps, but the space is basically the same. I'm going to put them all in the same jar. So we can call that big jar another bigger event that is containing the other 2 events that I just described. So now we have a big event with 2 sub-events, which are the ones that have the occurrences. And so the big event has now 4 occurrences, and within it we have 2 sub-events, right? So we can think about it like plots within a site. This is a typical ecological thing. And if we go bigger still or larger still, we can do that in all South America and grab all the sampling that was done in different countries, and we can call that an event that, of course, will have a spatio-temporal grain that is much larger. We're going from a little spot to all South America, and probably from a particular date to a range of dates. But we can also think about it as a bigger event with 2 sub-events inside, which were the sites, and those with sub-events inside of them. So you see that this is getting nested, right? So we have a hierarchy, and we have nested events one inside the other. And so basically we can think of a structure that is parent-child with the events, and we can relate each other with its parent or with its children. So with this in mind, what we want to do with Humboldt Core is to be able to represent all these levels of depth that one might want to show. So one might want to show at the plot level, at the site level, at the continental level, and all that should be possible. And so basically we worked with the paper, the terms that were proposed in the paper by Guralnik and collaborators, to build the terms that will be part of the Humboldt extension. And so this extension is currently under development. We're meeting every week or every 2 weeks. And so we're a bunch of ecologists and ontologists and standard experts that we're trying to figure out which are the terms that would help us capture all this information within all these different levels of complexity. And so the idea is to provide a minimum information vocabulary, that is this extension, to represent inventory processes, but also other levels within those inventory processes. So it has to be flexible enough to accommodate any kind of event that anyone would want to show or to share. And so we're basically grabbing the terms and reviewing and assessing whether those originally proposed terms are fit for this purpose. And we're organizing them in 6 loose categories. These are not classes as formally defined, but are categories. And so we have some terms that are general data set and identification, and we're grabbing some terms from Darwin Core to represent that information: geospatial scope and habitat, temporal scope and taxonomic scope, methodology description, and the part for completeness and effort. So I'm not going to go into the details, but just to show you an example of one of the terms, we have inventory type, which, as any Darwin Core term, as any standard term, will have its definition. We'll have some comments or not about its usage and examples of the values that one would expect under that term. We invite you all to join us in our meetings. You're most welcome to discuss with us all these terms. And so what we will do now is to finish revising all the original terms proposed for the Humboldt extension. As I was saying, reformulating the definitions and examples, and then we're going to test that extension with real data sets that we're gathering. So we're going to put it into the GBIF IPT and see what happens when we try to publish using this WIS extension. After that, we hope to ratify it through the TDWG process. So that goes through a public review and hopefully a ratification as an extension to Darwin Core. And an important piece is that we want to develop documentation on how to use this with different use cases. So if I have this level of nesting in my sampling, how do I use it? Do I use all the terms all the time? And the answer is already no, as you may imagine. There are some terms that are useful for some cases and some others for other cases. But as I was saying before, we intended to be flexible enough to accommodate the user's needs. And so we're trying to provide a solution to share this inventory data and other sampling data to improve its discoverability, interoperability, and reuse, and mostly also to lower the reporting burden, which is very high right now when one wants to put together information from different sources. So again, we invite you to join us. Here is our GitHub repository, and I want to thank everyone involved in the task group. This is a lot of work from a lot of people. So thanks everyone, and thank you all for listening. Now we'll take questions if we have time now.
Thank you, Paula. I'm pasting in the chat the link to our TDWG group. Okay, we have a question in the chat. Yeah, go ahead.
No, I was saying I was stopped sharing because I couldn't see the
We have a question in the chat from Carlos. Some institutions are already adding an expedition field to specimen records. Is that field already in Humble Core? I believe that it is related to your initiative.
Thanks, Carlos. We don't have a term that is expedition, and we have been discussing quite a bit how to accommodate, for instance, the people that are related to the sampling. And especially in cases where we have summaries, because those summaries involve so many people, because they are coming from different sampling events or campaigns or expeditions themselves. And so we are stepping back from adding this kind of information in Humble Core for now. However, we invite you to join us in that discussion. Darwin Core already has a term to capture who made the observation or collected the thing that is recorded by. And people sometimes I've seen it done in museum data, they use that field to capture expedition, blah, blah, with the name of the expedition, if they don't want to name the people in particular. But we're stepping back from the details of that. Shani, you want to add something?
No, I think your answer was very detailed. I'm hoping, Carlos, were you referring to that?
Yeah, I was referring to that. Many thanks. I was just asking because I think it is indeed related to what you are trying to standardize and record and capture. And also because, for example, it was common, and not just in the 20th century, but also in the 19th century, to publish a report on expeditions. And that opens the door to a lot of metadata that is often not in the labels, not in the institutional accession books, not in the catalog books. So that information is often what we are looking regarding target taxa and completeness and effort and how many people. So if we are trying to incorporate that kind of event information, then it can be useful to have an expedition field that captures the name of the expedition and might be useful for fetching additional data. I can help with that if the Humboldt core is open to dealing with the term or related information. I can try to give some input now and post that week.
Yeah, that will be great. Yeah, yeah. We invite you to join these groups. It's like open conversations and discussions every Wednesday. And yeah, and there's another comment related to the expedition about including information about permits and how due to the Nogosha protocol, it's very important to record that information. So yes, we welcome everybody to include these comments while we're developing the Humboldt core. It's like perfect timing to open this discussion. I think we should move on to Peter Brenton's presentation. Thank you, Peter.
Okay, can everyone see that all right? Sorry, my screen display has switched, so I'm not sure.
It's perfect.
Great.
Yeah,
okay.
It's perfect. We can see it.
Excellent. All right, so actually it's a really nice segue because from Yanina and Paula's presentation, because my presentation is really looking at an implementation approach that we're looking to adopt. We're already adopted part of it, I guess, and looking to formalize it and extend it further within our field data collection tool in the Atlas of Living Australia, in a product that our flagship field data collection tool called BioCollect. So hopefully this will work. So basically, I'll just an outline of the presentation. I'll try and keep it as brief as possible, but there's a fair bit of material to get through, so I'm hoping it'll get in on time. So we'll look at a few characteristics of surveys, some of the key differences in data concepts that exist within sort of relevant areas, the basics of ecological surveys, and then sort of dive into how we're actually implementing those Humboldt core terms as extensions to event core and Darwin core within BioCollect, and then finish with some summary thoughts. So looking at characteristics of surveys, basically a survey, I guess, is a deliberate visit to a place and time, or a set of places and time at a time or time period, to sample an area for what it contains. And typically it would have some sort of systematically applied method that may in fact be some sort of defined period of time where opportunistic observations are made, but nevertheless it is and an area covered, but nevertheless it would be some sort of systematically applied method. It may be more formal than that. Generally they're event-based, that is place and time context. And they typically would also involve things like covariates and measurements and things around phenology and weather and landscapes, geology and that sort of thing, in conjunction with the biota that is being recorded at a place. And they may also include some sort of collection of specimens or samples. So if you look at the data concepts, Darwin Core is a very flat record-centric standard. Everything basically revolves around an occurrence. So an occurrence happens at a place and time, as Paula mentioned in her presentation, and also Diana in hers. Often the the event results in a single record, with a record-centric approach, you could sort of make an assumption that independent records that share the same place and time collected by the same person are in fact collected in an event context, but that is an assumption and not an explicit thing. So an organism has an occurrence has an identification, a taxon is associated with an occurrence. There may be material samples, there's geological context measurements, and facts being essentially any other thing that's recorded. Humboldt core is an extension essentially to event core, and Yanni and Paula mentioned some of the key concepts that have been covered in that, being around methods, effort, and completeness, essentially as metadata, but can in fact be at the record level as well. I'll cover that in a moment. Another common way of looking at things is from a location context. GIS systems often look at things in this way. Everything's associated with a place. So a place has certain characteristics, biological, physical, geological, and so on. Observations of occurrences and other things are made at places. Events occur at places. Samples are collected at places, and so on. So samples might be associated with observations, measurements, and facts, of course, associated with various other things as well. But essentially this sort of way of looking at things is all location-centric. Integrating those concepts, we sort of taking an event-centric view, that is the visit to a place and time at a time or a time period that has certain attributes, and some of those will be observations and some will be other things. So the location, of course, can have its physical and biological characteristics, and the observation can be of organisms which have identifications and taxa and so on. Material samples can be associated with observations, and measurements or facts can apply to any of those concepts. So it's obviously a much more complicated sort of way of looking at things, but the real world's a complicated place. So there are a couple of working standards that are applying sort of these concepts. Obviously, in the biodiversity world, Darwin Core, Event Core takes this sort of view. Perhaps not quite as rich as this. It flattens it out quite a lot, but the PPSR core as a citizen science-based standard is also sort of modelled around these concepts. Our system is based around the PPSR core. PPSR core stands for Public Participation in Scientific Research. It's a relatively new standard and governed by the Citizen Science Association in the US, actually. So event data, just a few concepts around surveys, survey context. So use cases for event data, the species inventories, as Yanni mentioned, and also covariate analysis, which might include species. And it's the basis of a lot of ecological analyses. They can be really simple, but they can also be really, really complex. We've actually just implemented a really complex method that incorporates structural diversity of vegetation condition, vegetation composition, structural diversity, and also covariates measured against benchmarks for a particular ecosystem type within our system, and it's actually really, really complex. The species co-occurrence at a given place and time is really important. I think Diana mentioned that in her talk as well. Sampling method has been mentioned by a number of people in this talk, this session, sorry. Covariates can be really, really variable, and they're often related to the research questions that are being asked in any given survey. But it's important to capture that context with the records as well. Absences, as have a number of people have mentioned, are really important, and effort and comprehensiveness also. Site homogeneity is a really interesting one because scale, I think, is an interesting question that we still need to tackle, and that was raised in Paula's presentation. We'll come back to that in a little bit. And often replicated sampling techniques are used. Am I running out of time? Sorry. So a couple of challenges in this sort of event context data. Modeling, navigating, and visualizing structured, nested type of data, replicated sampling approaches is actually quite challenging. Yeah, it often requires a lot of computational power. So there's quite a few challenges with that. The other use case that's really important in this for this type of data is around time series, so temporal patterns and trends in populations and distributions, and often relative to covariates. So another reason why covariates are really important to capture. So obviously it requires two temporally separated data points at a place, and often, but not always, with compatible or comparative methods, so that you've got a measure of relatability between the two data points. Absences become really important when it comes to temporal analysis, and also the concepts around comprehensiveness and completeness of survey effort or survey coverage. But the challenges around what is a site when you're looking at size, how close in proximity do measured locations actually, or sampled locations, need to be in order to be comparable for temporal patterns, is a really challenging thing from a databasing point of view, and potentially from an interpretation point of view as well. How many replicant temporal points do you need to actually be statistically valid? Particularly in Australian data, we've got something like 100,000 data points, 100 million data points in our database at the moment, but they're very, very sparse temporally and spatially. So, getting enough data to be able to provide reasonable temporal analyses at scale is challenging. So, and there's also research data questions. So a quick look at BioCollect. This is, as I mentioned, our flagship field data collection tool. It services citizen science community, applied ecology, industry sort of sector, the research ecologists, and also natural resource management sector. So these, it's quite broad. We've got about bit over 3,000 projects in there at the moment, over 1 million presence and absence observations, and services a really wide range of the basically all the sectors that interact with biodiversity in some way through government and private sector. The entity structure in BioCollect basically revolves around a project, and projects can have one-to-many surveys. Surveys basically represent a particular protocol, and several projects. This one is actually a citizen science project that's running, that's doing hormonal analysis from scats as well as observations. So it's a multi sort of survey thing, but each one is different in what it's trying to collect and the methods that apply. And the data set, the metadata is defined at the survey level, and it's represented by a data schema, i.e. a form, and multiple usages of that form produce records. The records make up a data set. In terms of the survey metadata, we currently have a reasonably comprehensive schema that still needs further extension now with the work on Humboldt core. So some of the things that are sort of there at the moment: survey method. We have cover, protocol description, protocol name, protocol references, and that sort of thing within the metadata schema. We also have temporal spread for a particular survey, and represented by the first record in the data set and the most recent record in a data set, which may be the last, or it might be just the most recent in an ongoing data set. There's also a bunch of other information that's covered in the Darwin Core Standard around license and ownership, and a couple of extra things around quality assurance and data management. But then there's all these other attributes.
Peter, we have 3 minutes left for the session. So yeah, thank you.
Thank you. So, so a few things that aren't in there are things around effort, and at least at the metadata level, aggregated effort, spatial scope, temporal taxonomic scope, and so on, abundance measures, and whether samples were taken or not. Some of these can actually be derived from the data records themselves, so we can pre-populate those from the data, the event records that are created. The Humboldt Core team's also been looking at survey methods and trying to classify, quantify survey methods. So these group into some logical groupings around passive methods and sensor-based and non-sensor-based, and with sort of the taxonomic attributes and so on. I won't go through all of these. You can have a look at the slides afterwards, but there's a lot of them. We've tried to, rather than actually name particular methods and all the variations, we've tried to sort of clump them into groupings that use similar type of, sort of aggregate objective things, so that information is compatible or can be compatible within particular variants of them. The survey, the event form, within our structure, we can record event level information. So it's the place, time, collectors, and so on, and record level information. I don't know how clearly that's coming through to you, but that's a list of taxa. This is a macroinvertebrate survey that's done using a systematic method called Signal 2. This is how Humboldt Core terms map into this. So we've got things like the event time and temporal scope, event duration, so the amount of time that people are actually on site doing sampling, the methodologies that we use to actually collect, and also the habitats that were sampled as part of that particular survey event. There's obviously verbatim site names and location, and then there's, that's actually not showing the right thing, but it's supposed to be pointing to absent taxa. So these are taxa. This method sort of samples about 45, 43 different taxa. And so every visit to a place and time will generate absence records. It'll also generate presence records. We can also do a species abundance calculation directly from that data. So, the schemas vary wildly depending on what people are trying to collect, and what we're trying to do is design it in a way where we can actually map the attributes that are common across the different survey methods to the standards. So at the data schema level, we can do mappings to Darwin Core, so it's recorded by, event date, event time, scientific name, and so on. And then we can do aggregations of common attributes across different surveys for analysis purposes. So in summary, biodiversity captures a lot more than just individual records. Many systems actually focus on individual records, but that leaves out a lot of really relevant and useful information for analysis and context setting, and limits the usability of the information for some of those other use cases. Methods, effort, and completeness, and context in which information is collected is really, really important, both for interpretation, comparability of different data sets. Some methods are not compatible If you've got a fauna survey of an area, for example, that's done at night time using spotlighting, it will be completely different and not really very compatible with a pitfall trapping approach in the same area that will happen during the day or perhaps overnight. Covariates are important. They'll be different across different surveys, and it's important to be able to compare those across different surveys as well if you're looking at time series analysis and so on. In terms of databasing requirements, navigating and visualizing the nested data, we need to work out really effective ways of being able to do that. It's a tough problem. The metadata is really important and needs to be documented in a way that's actually sort of fairly standard so that it can be navigated effectively. And yeah, lots of issues to resolve. What's the site? You know, how to scale up to include all of these covariates with the vast amounts of additional data we'll get with absence records. How to display absence records in conjunction with presence records is another interesting problem. So, thank you.
Thank you, Peter, and thank you everybody for staying a bit longer. We obviously ran out of time, but thank you, Peter. That was a great presentation, and I think it's a good ending of the session, right? Like an implementation of these standards that we're generation with a very complete survey protocol and an interface. So I think it was great presentation. Sorry, I got lost in the Q&A part. There's a couple of questions, but I'm also realizing that it's already past 8:30. Well, whatever hour it's in your country or location. So I welcome everybody to join our meeting groups about Humble Core, and everybody is welcome to also include more questions and contact directly with the speakers. Paula, do you want to say something else?
Well, I think for those who stayed, I think we can stay a couple of minutes more because there is no other session here. And indeed, Abby was saying here that she thought it was half an hour longer. So I think we can stay a couple of minutes if you want to grab some questions. I would not want to leave, however, without thanking the TDWG organizers, the organizers of the conference and the program committee that have done an amazing job to put all this together. So thanks to all of them. So we have a little more time. So if you wanna pick questions, Shani from the Hula. There are a couple that I have been answering there. But there were also some questions for, let me see, for some of our first speakers. I can't find them now. It's something where I can only see 2, and I know there are 7 unanswered.
There's an interesting question regarding if it's safe to assume absence in inventory and survey data if a species is not recorded. The question is larger, or must you specifically say this species is not here, and should we call it absent or not detected? This is very important, like a key issue in inventories, right? And the fact that we can provide information about the target. Sorry. Okay. Sorry, I thought I was muted. If we can provide information about the targeted species, so the list of species that we assume for an area and which ones are not detected, and according to the sampling methodology and the effort. Of course, we can never assume that there will not be any false absences, but we are certainly closer. Regarding if it's absent or not detected, that's a good question. We are including the term as absent taxa, so we're including absence there. But it could be a good discussion. Maybe I don't know if Rob Borelnik or Peter want to say more about this.
I think it very
Peter.
much depends. I think it really depends on how explicit the method is in terms of the target species. You know, if you're going to an area specifically to target certain taxa and you don't find them in a particular sampling event, then it's probably, you know, fair to say that they were absent given the, you know, the constraints of the effort that was expended in sampling that area and the assessment of how complete that sampling was. But you know, it is sort of really dependent on you know, those factors, because, you know, if you didn't spend very long sampling an area, even if you went with intent to sample particular taxa, you're probably more not detected rather than genuinely absent.
I see a question here in the chat that I would like to pick up. That is, Deborah Piñatari is asking, how are we planning to have a comprehensive contributions to the test phase? And that's a very, very good question. And I want to take the opportunity to invite you all who are here in this symposium to participate in that test phase. The idea is to have some data sets from groups or use cases that we already know, but we're going to contact different groups so that they can provide data sets. The idea is that the first test has to be with something that we know how it works and not just with random things that come from outside, because it's very difficult to actually test that it works. But after that, I think we want to open that testing phase to anyone who wants to use it and see what happens, and we can receive comments and suggestions and changes, etc. But it's going to be a very important phase, and I invite you all to participate when we let everyone know that that is ready.
I would say, like, in the last 5 minutes, if anybody has a question, just open your microphones and speak. This is the moment for that.
Hi, this is Rob Stevenson. I just
Hi,
wanted
Rob.
to comment, on the importance of whether data are, whether you know there's an absence or not. That will presence absence versus some kind of effort collected data, and that difference will the importance of that difference depends on what you're trying to do with the data. If you are interested in a some things like a range map, it might not be so important, but if you're trying to get population density, it would be very important. So I think it's the place to look if people are interested is in the modeling that people do and the analysis of the data. The models will clearly show how to model if you're interested in estimating population versus a range map.
Thanks, Rob. Sorry, I was muted. Does anyone want to jump in with questions directly? Many of the questions that were posed in MOVA are answered there already. Finding any unanswered one except from one from Annie Simpson asking why are we calling this humble extension? But that's for historical reasons, I think. That's what they call it and what they propose as a humble core. And Rob Browning and collaborators. And if someone else is very annoyed by the name, we may consider it, but for the time being, it's going to be called humble extension.
I like the humble part. Any last minute questions for any of the speakers present?
I'll ask some. This is Deb.
Hi, Deb. Go ahead.
Hi. I'm curious about a couple points anyway that we can continue the conversation. So over the years, there've been a discussion about identifying grants associated to a given project, or in this case, I was thinking about expeditions, and where would that number go? And it occurred to me that that might mean an expedition would have a unique identifier itself. And then I thought about this going back in time versus forward in time question. So if we go forward in time and look back at the data that's being collected in Humboldt Core, is it what the ecologists of the future need to look? And how does that relate to the concept of a voucher? So there's a thematic collection network in ADBC right now that is looking at marine invertebrate museum specimens. And of course, inside a jar with a coral in it, inside that coral, you can see all these other little critters that are living on the corals, epibionts, right? And so they're trying to capture some of that information as well. So it's like going back and doing that sampling, that marine sample, and sort of recreating, you know, who was there in that sample, what was present, if you will, in a very, well, in a unique way, I would say, right? Going back in time to recreate that event. So does Humboldt Core have a way to connect vouchers to, or that there's information or vouchers included in the sampling method? So two questions: identifier for the grant and vouchers.
I would say none of the two. Identifiers for the grant, I think, and this is related to what someone was asking before about expeditions. This is more related to the people that are involved, and we are somewhat on purpose staying out of that. We have discussed it, and we will probably discuss it 10 times more before this goes to public review, even for testing. And I think that's probably something that we have to talk with the other group at TANWI who's looking at people, how to capture that information. About the vouchers, I think we're touching there. I mean, this humble extension is to capture information about the inventories, but we cannot forget that this is tied to an event, and that event is tied to occurrences, probably, if we want to tie them, or if whoever is publishing wants to do that. And so the voucher part would come, all the information about the voucher would come attached to the occurrence part of the thing, and not to the humble extension itself. So we did discuss, however, a term that has voucher or not. And we decided to leave it out, I believe, if I'm not mistaken. We decided finally to leave it out because that's on the occurrences side of things. And so you already have that information coming with the record or associated to that record somehow.
So the record would say that this voucher was collected as part of the expedition, the occurrence record.
If you have the occurrences linked to that event, yes.
Yes. Okay. I mean, I was just thinking that, like I said, the presentation that was given yesterday somewhere about the recreation, like of expeditions, like the HMX expedition, and the specimens are scattered all over the world, right, in collections, and recreating what we know about that expedition from the vouchered specimens. So I was trying to imagine 100 years from now, right, what we would do. Thanks for that, Paula.
In a couple of years from now, for those of us who have been following the latest GBIF news, the whole publication schema is going to change. So we will see how we adapt this to new ways of publishing data in the very near future. So we might have to revisit many of these discussions and many of these ways.
Yeah, my thought was that oh, I'm sorry. Did you?
Go ahead,
I was thinking. Okay, I was thinking that the this notion of presence and absence and the certainty and those kinds of things would change as the events change with the collections of things like eDNA and the vouchered specimens. So your certainty and your methods, right, would potentially be affected by how, whether you want to say it's not detected versus absent. Yeah. Other peeps must have questions besides me. Yeah.
I'm seeing that at some point we had like 195 attendees. So thank you everybody for joining. This is great. And thank you for having interest in inventory data and biodiversity standards. So I don't know. I think this was amazing.
Indeed. I invite you all to write to us or push some issues in the GitHub repository if you have them. And of course to join us in our meetings. Just shoot us an email or a message, and we'll be happy to have more people involved. And thanks to all our speakers today. It was great to see different sides of the thing, different elements that we definitely need to connect better.
I think what this shows us is that connection between different groups and different people's working on biodiversity and data collection. It's key to solve many issues, or at least to try to solve these issues. So yeah, thanks to everybody for your questions, for your presentations, and hope to see you well again, obviously in all this week in TAD Week, which has amazing presentations all the week long, and also in our working group. So everybody's invited.